Good day, everyone. I am Tom Rapada, and this is Jasmine Televis. Today, we are going to discuss about oogenesis in a female cow and the cell division anomaly called non-disjunction. So this presentation will explain how egg cells are formed in female cows, why this process is important, and how errors in cell division can lead into abnormalities.
Cell division is an essential process in all living organisms. It allows growth, reproduction, and the production of specialized cells. In female animals, including cows, a special type of cell division called meiosis produces egg cells. This process is also known as oogenesis.
Our chosen organism is the female cow. Its scientific name is Bossaurus. Cows are mammals that play an important role in agriculture by providing milk, meat, and offspring. Like other mammals, female cows reproduce sexually and produce egg cells through the process of oogenesis.
The diploid chromosome number or 2N of a cow is 60 chromosomes. The haploid chromosome number or N is 30 chromosomes. The oogeneum and primary oocyte are diploid cells because they contain 60 chromosomes. The secondary oocyte, polar bodies, and mature ovum are haploid cells because they contain only 30 chromosomes.
Oogenesis is a process by which female reproductive cells or egg cells are formed in the ovaries. This process involves meiosis, a type of cell division that reduces the chromosome numbers by half.
The first stage begins with oogonium, which is a diploid cell containing of 60 chromosomes. The oogonium grows and develops into a primary oocyte. The primary oocyte then enters meiosis 1. During meiosis 1, the primary oocyte divides into two cells, a large secondary oocyte and a small first polar body. The secondary oocyte undergoes into meiosis 2 and produces a mature ovum and the second polar body. At the end of oogenesis, only one functional egg cell is produced, while the polar bodies eventually degenerate.
Primordial follicles in the ovaries contain primary oocyte cells. As follicles mature, primary oocytes become secondary oocytes. These are the egg cells produced by oogenesis. During ovulation, a follicle bursts and releases its egg from the ovary.
Oogenesis is important because it produces the female gamete needed for reproduction. It also reduces the chromosome number from 60 to 30, ensuring that the correct chromosome number is restored during fertilization. In addition, meiosis creates genetic variations which help populations adapt and survive.
So our chosen cell division anomaly is non-disjunction. Non-disjunction occurs when chromosomes fail to separate properly during meiosis. As a result, some egg cells may receive too many chromosomes, while the other may receive too few.
Normally, homologous chromosomes separate during meiosis 1, and sister chromatids separate during meiosis 2. However, during non-disjunction, these chromosomes do not separate correctly. This produces abnormal gametes with an incorrect chromosome number. When these abnormal egg cells are fertilized, the offspring may develop chromosomal abnormalities.
So the effects of non-disjunction may include infertility, developmental abnormalities, reduced survival of embryos, and pregnancy loss. In cattle, many embryos with severe chromosomes abnormalities do not survive to birth. So, therefore, accurate chromosomes separation is very important for normal development.
There is no guaranteed way to prevent non-disjunction because it occurs naturally during cell division. However, proper breeding management, genetic screening, and veterinary monitoring can help identify reproductive problems.
So, in conclusion, oogenesis is the process by which female cows produce egg cells through meiosis. The diploid chromosomes number of a cow is 60, while the haploid number is 30. This process is essential for reproduction and genetic variation. We also learned that non-disjunction is an abnormality in cell division that causes chromosomes to separate incorrectly, leading to abnormal chromosomes numbers. Understanding both normal and abnormal cell division will help us appreciate the importance of genetics in reproduction and development. Thank you for listening.